Yes, welcome back. In this fourth video of this chapter, Cell, the Unit of Life, I am going to deal with the last two important organelles, mainly the chromosomes and the cytoskeletal structures that forms the cilia, flagellum and the centrosome. So, now I am going to take the structure and function of the chromosomes. Now, chromosomes assumes different shape at different stages of the cell cycle. In the next chapter, we will be learning the different stages of the cell cycle, namely interphase, prophase, metaphase, anaphase and telophase. At the various stages of the life cycle of a cell, see, the cell undergoes different stages in its cell cycle. The first phase is the interphase, next is the prophase, then metaphase, anaphase and telophase. The shape of the chromosome changes, but in textbook we usually give reference to the interphase stage as well as metaphase stage. Interphase stage of the cell cycle, the chromosome appear as fine coils, entangled network, and it is referred to as chromatin fibers. So, reference is given to the interphase stage where it appears like a reticulum or it appears like a loose network called the chromatin fibers, or it is made with respect to the metaphase. Metaphase, it appears like X-shaped. Okay, so the next lesson we will be learning how it is getting these shapes. Okay, so now to explain the different types of chromosomes, I have to give the diagram in context to metaphase and description in context to prophase. Now the same thing if I show it in prophase, it just rod shaped. So we have to remember that the chromosome, when it is in the interphase, it is entangled network. Then as it proceeds to the next phase, that is prophase, what happens? This entangled network condenses or shrinks and therefore becomes rod like thin rods. Again it further shrinks and becomes X-shaped, thick X-shaped structures. Okay. So with reference to these three is given in textbook. When they talk about chromosomes, they either give, usually it is in the interphase phase or in the metaphase stage. Clear? So now I am going to explain the structure of the chromosome with respect to the metaphase stage. So this is I, interphase, this elongated rod like structure is the prophase, and this X-shaped structure is indicating the metaphase. Clear? Now describing the chromosome in its metaphase is that it has a constriction, and this constriction is known as the primary constriction or centromere. See, a primary constriction is seen. Now I have located that constriction at the center, and because it is located at the center, it is seen like an X with a central constriction, and that constriction is called primary constriction or centromere. In some chromosomes, there is a secondary constriction. I am going to show the secondary constriction on the last chromosome. See, secondary constriction, such that it gives the appearance of a small segment attached to the chromosome. See, it looks like a small segment attached to the chromosome. This is called the secondary constriction, and this secondary constriction is called satellite. And when we stain it, it does not take up the stain. So once again, the chromosome appears in different shapes according to the stages of the cell cycle. The 5 stages of the cell cycle are interphase, prophase, metaphase, anaphase, telophase. In the interphase, it is like an entangled network called chromatin threads. As we proceed to prophase, it condenses and becomes rod-like. Further condensation becomes X-shaped. With respect to this metaphase chromosome, I am describing the two constriction. The primary constriction is called the centromere. Usually it is located in the middle. And there is a secondary constriction only in some chromosome, giving a small fragment attached to the chromosome. Usually this does not take up the stain when we stain it, and this is called the satellite. Now, with respect to the location of the primary constriction or the centromere, chromosomes can be divided into 4 types. Metacentric chromosome, where the centromere is located in the middle. Only I am concentrating at the location of the centromere or the primary constriction. In metacentric, the centromere or the primary constriction is located in the middle. In submetacentric, it is slightly away from the center. The centromere is slightly away from the center. Acrocentric, the centromere is towards the tip, very close to the terminal end. And telocentric, the centromere is located at the terminal end. So, I emphasized only one point, location of the centromere. Here in metacentric, where is the centromere located? In the middle of the chromosome. Whereas submetacentric, it is located slightly away from the center. Acrocentric is somewhere close to the terminal end, and telocentric, it is at the terminal end. So, because of its location, how is the arms, the size of the arm? When it is located at the center, it divides the chromosome into 2 equal arms. So, this arm is equal and this arm is correctly equal. It divides the chromosome into 2 equal arms. But in metacentric chromosome, it's slightly away from the center, the primary constriction. So, it divides it into a very long arm and very short arm. A long arm and short arm. Then, in acrocentric, it is located almost close to the tip. So, it divides it into very long. Here you must use the word very long arm and very short arm. And in the telocentric chromosome, because it is located at the tip, there is only lengthy one arm. And when I am talking about arms, you will be wondering I am talking about one arm this side and one arm that side. It is, when we are describing with respect to prophase. So, prophase, I am locating the centromere, prophase stage. It is located, see, centromere is located in the middle of the chromosome, dividing it into 2 equal arms. Then submetacentric, it is slightly away from the center, dividing it into 1 long arm and 1 short arm. Then acrocentric, the centromere is close to the terminal end, divided into very long arm and very short arm. And telocentric, it is located at the tip. So, with respect to the prophase, you are giving the description, but you are drawing with respect to metaphase. I told you about the phases. This is the description with respect to prophase, and the diagram is with respect to metaphase. Clear? So, all you need to remember is the location of the centromere and the size of the arm. So, once again reiterating the point, metacentric, the centromere is located in the middle, dividing the chromosome into 2 equal arms. Submetacentric is slightly away from the center, dividing into long arm and short arm. Acrocentric, little towards the tip, and so it is dividing the chromosome arms into very long and very short arm. And telocentric, the centromere is located at the terminal end. The function of the chromosome, next aspect, function. We know they are the carriers of hereditary information, so they are involved in inheritance. They transfer information from the parents to the offsprings. So, these chromosomes are tightly packed with, see, packed with DNA. And the DNA are held, see the packaging, highly coiled. Their DNA are held with some proteins called non-histone proteins and histone proteins. These are basic proteins. So, these coils should be packaged, or else it will get uncoiled. So, this packaging is with the help of proteins called histone proteins and non-histone proteins. These are basically basic proteins, not acidic proteins. So, they are tightly packed. So, the chromosome, if you analyze the chromosome under the high power of the electron microscope, it is made up of DNA, and DNA is highly coiled and packed with the help of histones and non-histone proteins. Clear? Next, organelle. These 2 organelles, cilia and flagellum. First I will deal with the function. We know definitely both of them have the same function. And what is the function? Locomotion. They enable locomotion, motility of the cell. And the very example that strikes in you is flagellum in Euglena. See the flagellum. and the cilia present in paramecium. Okay, very two good example to make a comparison, two protistants, euglena and paramecium. Euglena, it moves with the help of flagellum, and paramecium moves with the help of cilia. And the difference in structure is cilia is short, very short, and they occur in numerous, the number numerous, and they occur in a row. Whereas flagellum, they are single and very long. Usually single, there are some organisms which have two flagellum. Okay, anyway, they are long thread-like structures, whereas these are short and they are numerous and occur in a row. Now, the way they move, this move like in an oar-like fashion. You know the oars of the boat, this one, or not that all the cilia move together. It's the few cilia will be moving like this. So, in that oar-like manner, if you observe the movement of the boats with the help of oar, you can see all the oars do not go together up in an oar-like manner. Okay, there is a coordinated motion. Whereas this shows a whip-like motion. You see in the whip how it moves, the whip-like motion. Now, that is dealing with the differences between cilia and flagellum. Both of them are involved in locomotion. When you take an electron microscopic view of the cilia and flagellum, or the ultra structure of the cilia and flagellum in cross section, this cilia and flagellum, they are not jelly like, or they can be stiff, or they are supported by cytoskeletal structures. So, this cilia and flagellum are supported by cytoskeletal structures. So, next we will study about the cytoskeletal structures and come back to cilia and flagellum to give the arrangement. Okay, so but now I will reveal the arrangement of the cytoskeletal structure. They are arranged in this arrangement, 9 plus 2 array. So, remember the cytoskeletal structures are arranged in the 9 plus 2 array. So, once again, before I move out from the slide, let me explain cilia and flagellum. The function is locomotion. Most of the protists are provided with cilia or flagellum. A very good example, euglena with flagellum and paramecium with cilia. The basic difference between cilia and flagellum, this flagellum occurs in 1 or 2 numbers, whereas cilia numerous. Then flagellum is long and thread-like. There it is short, hair-like structures. This move in a whip-like motion, there it moves like an oar-like fashion. These are the three differences between cilia and flagellum. Then we will take a cross section and view it under the electron microscope, or the electron microscopic view, or the ultra structure of the cilia and flagellum shows 9 plus 2 array. The meaning of 9 plus 2 array is the number of microtubules that supports the cilia and flagellum. Clear? Remember, it is 9 plus 2 array. Next, centrosome. Centrosome, as I told you, this is also a non-membrane bound organelle found in animal cells. Okay. Now, talking about the structure of centrosome. A centrosome consists of two cylindrical structures. See, I am drawing one cylinder, and look at the other cylinder, second cylinder. So, how are they located? They are located at right angles to each other or perpendicular to each other. So, it comprises two cylindrical structures, short cylindrical structures which are located perpendicular to each other or at right angles to each other. Each cylinder is called centriole. And remember, it is not surrounded by a membrane. So, I have to cut off this membrane. It is not surrounded by a membrane. So, once again, what is a centrosome? Centrosome is found in only animal cell. It consists of two cylindrical structures called centrioles, and the orientation of the centrioles, they are at right angles to each other or they are perpendicular to each other. And now, this also under the electron microscope or the ultra structures shows 9 plus 0 array. That means the arrangement of the microtubules within it is 9 plus 0. The previous cilia flagella, I told you 9 plus 2 array. This is 9 plus 0 array. Okay, we will analyze that in the next template. Now, what is the function of the centriole or centrosome? They form the basal body for three structures: cilia, flagellum and spindle fibers. So, once again, what is the function of the centrosome? They form the basal body, okay, the basal body for cilia, flagellum or spindle fibers, clear? The base or the basal granule for these three structures. So, once again, before I could move to the next template, the structure of the centrosome, it comprises two short cylindrical structures. What are those cylinders called? Centrioles. And how are they oriented? At right angles or perpendicular to each other. And the centrioles under the electron microscope shows the 9 plus 0 array. Okay, this we will understand in the next template. Now, the function of the centrosome or the centriole, it forms a basal body for cilia, flagellum and spindle fibers. Next, yes, this is it, the cytoskeletal structures. See, cilia, flagellum, spindle fibers, centrioles, basal bodies, etc. Though they are thread-like structures, they are supported by a skeletal structure. The structure is the form of tubules or in the form of fibrils. For example, this pen, it is stiff. Why it is stiff? Because it is supported by this wall. But in cilia and flagellum, there is no wall to support. There are some cytoskeletal structures that should support it. For example, this is the cilia inside what? If there is no cytoskeletal structure, it becomes very flaccid, jelly-like. Okay. So, there are these cytoskeletal structures. Take these as cytoskeletal structures. The cytoskeleton, microtubules or microfilms, they are placed inside. So, it provides support to the cilia. Clear? So, fixed. Now, it has become very rigid, very strong. Okay. It is not flaccid, flexible because of these cytoskeletal structures or tubules. The cytoskeletal structures can be in the form of microtubules or microfibrils. See, if I am adding more, you can see. Okay, I am adding more, and this is providing the strength, rigidity to the cilia or flagellum. Clear? Now, how are these arranged? So, talking about the arrangement, it is not just a bundle of microtubules, just not a bundle like this way, like how we hold a broom, a bundle of stick. It is not like that. There is a specific arrangement. In the case of cilia and flagella, definitely there is the plasma membrane surrounding it. So, there is a plasma membrane surrounding the cilia or flagellum. Now, the microtubules, I told the skeletal structures are the microtubules. Now, how are the microtubules arranged? It is not arranged like this way, like a bundle. It is arranged in a specific manner. Now, let us see the arrangement. There are totally 20 microtubules. So, see how many of these I must have? 20 in number, but not held as a bundle together. They are specifically distributed. There are 9 pairs at the periphery. 1, 2, 3, 4, 5, 6, 7, 8 and 9 pairs at the periphery. So, when you take the cross section, they appear circular. So, there are 9 pairs of microtubules at the periphery. These 9 pairs are called doublets. So, what are these called? Doublets. So, 18 are there. Then 2 in the center. The 2 which are in the center, they are isolated, they are not close like a doublet, they are not in pair, but they are connected by a bridge. But they are connected by a bridge and enclosed by a central sheet like a bracket. I am showing central sheet. Okay. Then, very importantly, these 9 doublets are connected to the central sheet by radial spokes. But only one of the microtubules, so there are 2 microtubules, so 1 microtubule, 2 microtubule which makes a doublet. Only the second microtubule of the periphery is connected to the central sheet. Got it? So, how many, and this connection is called the radial spokes. Clear? So, there are 9 radial spokes. Then, with another color, say black, the doublets themselves are interconnected. See, this doublet is connected with this. This is connected with this. This is connected with this. So, the doublets themselves are interconnected by linkers. So this is called linker. And most importantly of all, what is missing? I have to draw the plasma membrane. Very, very important. Plasma membrane should be drawn. So once again, talking about the ultrastructure of the cilia of flagellum. Remember, it is supported by cytoskeletal structures called microtubules. See, and these microtubules does not occur like a cluster, like a bundle. It's occurring in specific orientation. So first, starting from out to in, the cilia of flagellum is surrounded by a plasma membrane, which is indicated in green here. Then there are totally 20 microtubules. 18 of them are arranged in the form of doublets, and these doublets are occurring radially peripheral at the periphery. So where are the 18? They are arranged in pairs, and these pairs are called doublets. And where are these doublets located? At the periphery, and they are arranged radially. Okay, see, you can see the first doublet, second doublet, third, fourth, fifth, 6 doublets, 7 doublet, 8 and 9 doublets. So like that way there are 9 pairs of microtubules called doublets, which are arranged peripherally and radially. Clear? Then there are 2 doublets at the center. So 18 is the 19th doublet and this is the 20th doublet. The 19th and 20th doublet does not occur in a pair, but they are connected by a bridge. See what is given in green, and the enclosed, see this red color, by a central sheet. Now the 9 pairs or the 9 doublets which are located at the periphery are connected to the central sheet by the spokes. What are these spokes called? Radial spokes. So how many radial spokes are there? There are 9 radial spokes. And last of all, the doublets themselves are interconnected. See, if I give it in a color that stands out, they are interconnected. One microtubule of the doublet are interconnected with each other. See, they are interconnected, and they are called the linkers. I hope you have understood. So once again, the ultrastructure of the cilia of flagellum is surrounded by the plasma membrane, and the central core, the remaining part of the cytoplasm, is called the axonema. In the axonema, the 20 microtubules are located, but the location is 9 pairs of these microtubules called the doublets are arranged at the periphery. So 9 of them arranged at the periphery and 2 at the center. The 2 that are occurring in the center are not close, but are connected by bridge, and they are enclosed by a central sheet. Now, one of the doublet of the 9 peripheral doublets, one of the microtubule are connected to the central sheet by radial spokes. So like that way there are 9 radial spokes, and the doublets themselves are interconnected by linkers. I hope this 9 plus 2 array is clear with the diagram. Clear? So now this is the view of the 9 plus 2 array. See, if we open it, you can see this is the distribution of the microtubules inside the axonema of a cilia of flagellum. This is only providing rigidity, strength to the cilia and flagellum, and also enabling motility. Clear? So that's the purpose of cytoskeletal structures. They give support to cilia, flagellum, and in the next template I am going to show is the centrioles. Clear? I hope the 9 plus 2 array is clear. Next from this you can understand it is going to be the 9 plus 0 array. Now here you can see 27 microtubules. But these 27 microtubules are arranged in the form of triplets. See, one microtubule, second microtubule, third microtubule. So like that way, 9 triplets at the periphery are located. So like that way, 3, 3, 3, 3, 9 times 9 3s are 27 microtubules are located at the periphery. Then the other description. At the center, there is no microtubule. Instead, at the center, there is a proteinous mass called hub. What is it called? Hub. H-U-B. Hub. And just like the previous case, the triplets, there the doublets are connected to the central sheet. Here, the triplets are connected to the central sheet by radial spokes. See? Radial spokes. So, one of the microtubule of the triplet is connected to the hub by radial spokes, and just like the previous one 9+2 array, the triplets are interconnected by linkers, and the difference is that there we had a plasma membrane, but here there is no plasma membrane surrounding. See, I am erasing the plasma. So, that is the difference between the 9+2 and the 9+0 array. 9+0 array, there is no plasma membrane, whereas 9+2 array, there is a plasma membrane. Next, here the microtubules are arranged in the form of triplets at the periphery. There they arrange, the microtubules are arranged in the form of doublets at the periphery. Then third difference, here in the center there is no microtubules, instead a proteinous mass called hub. In that case, there is two microtubules at the center, not in a pair, they are there isolated, connected by bridges. So, these are the three differences between 9+2 array of Cilia and Flagellum and 9+0 array of centrioles. The three difference, once again reiterating the difference in 9+2, if I write it side by side. 9+2, the example is Cilia and Flagellum, and 9+0, the example is centriole. Now, giving the difference between these two. So, first difference, plasma membrane is absent, is not there in the 9+0 array, whereas here plasma membrane is present surrounding it. Second, here how many microtubules are present in the 9+0 array? There are totally 27. How are they distributed? 9 into 3. That is, they are distributed in the form of triplets at the periphery. Whereas in Cilia and Flagellum, there are totally 20 microtubules. How are they arranged? As doublets. 9 doublets at the periphery and 2 isolated microtubules at the center connected by bridge. And the third difference, the center, is there a microtubule in the center in the 9+0, that is why it is 0. Center, there is no microtubule. So, no microtubule in the center. Here there is 2 microtubules. There are 2 microtubules at the center, but isolated, connected by bridge. Here instead what is it? It is a proteinous mass called hub. So, you got the three differences between 9+0 and 9+2. 9+0 is found in centrioles, whereas 9+2 is found in Cilia and Flagellum. First difference, no plasma membrane surrounding. There is plasma membrane surrounding. Second, there are 27 microtubules that supports the centriole, but the way they are distributed is in the form of triplets at the periphery. There are 29 pairs or doublets at the periphery with 2 isolated microtubules at the center connected by a bridge and enclosed by a central sheet. And the third difference is in the center there is no microtubule, whereas in 9+2 center there are 2 microtubules. Clear? And the similarities of both of these, there are radial spokes. 9 radial spokes in 9+0 and 9 radial spokes in 9+2 arrangement. What are these radial spokes? They connect the microtubule of the periphery to the center. And also another common feature in both is there are linkers. What are linkers? Linkers interconnect the microtubules which are at the periphery. Whether it is a triplet or doublet, they interconnect it. Now, when you look at this, so that is the similarities. Linkers are the similarity. One similarity is with respect to the linkers and the other similarity is with respect to the radial spokes. So, leaving aside all this, looking at this diagram, what appearance does it give? There is another name for this 9+0 array. This gives the appearance of a cartwheel. So, 9+0 array is also known as cartwheel. The cartwheel. Have you seen the bullock cart or the ox cart, the cart driven by the bulls? it has a cartwheel appearance. So, the 9+0 array is also known as a cartwheel array or arrangement. Clear? I hope you have understood. The microtubules, as I told you, the microtubules provide rigidity, strength, support and motility to structures like cilia, flagellum, spindle fibers and centrioles. They are not arranged like this way as a bundle, instead they are arranged either at the periphery like this way, at the periphery as doublets or at the periphery as triplets. See? Clear? So, with that I come to the end of this chapter. Thank you.